Hello, and welcome to the webinar on Improving the Safety and Quality of Organic Leafy Greens by Sadhana Ravishankar of the Department of Veterinary Science and Microbiology at the University of Arizona. My name is Alice Formiga, and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. You can find all of our published articles, videos, and our many upcoming and recorded webinars on organic farming and research topics on our website at extension.org/organic_production. This presentation is being recorded, and you'll be able to find the recording on our website and on the eOrganic YouTube channel within the coming week. Today's presentation will last approximately 50 minutes, and then we'll have an additional 30 minutes for your questions. If you have a question at any time during the presentation, you can type it into the question box and hit return. If you can't see the question box, just click the small plus sign next to the word question on your control panel, and that will open it up. We'll be reading the questions out loud after the presentation is over, and we'll answer as many as we have time for. Later today, we'll also be sending out a short evaluation survey by email. We're very pleased to welcome Sadhana Ravishankar, as well as Kurt Nolte of the University of Arizona Extension, who will be joining us during the question and answer period. Dr. Sadhana Ravishankar is an assistant professor at the Department of Veterinary, Veterinary Science and Microbiology at the University of Arizona. Among many other things, her current research focuses on controlling foodborne pathogens using various technologies and natural plant antimicrobials, improving the safety and quality of organic leafy greens, and understanding the survival of foodborne pathogens on fresh produce, composts, and compost teas. Kurt Nolte is the Yuma County, Arizona Extension Director and the Yuma Agricultural Center Director, as well as the Regional Vegetable Production Specialist. So before we begin, we would like to get an idea of who is online with us today. For example, how many people are organic farmers, and whether you sell organic produce, and whether you are a consumer of organic produce. So we're just going to pull up a couple of very quick polls, and we're only going to have them up here for about 20 seconds or so. So if you're online with us, you could just quickly check one of the answers here, and we'll have it up for a couple seconds and then move on to the next quick poll. So let me just share the results here for that. We had some certified organic growers, as well as some other growers who use organic methods but are not certified, and then some of our audience are not growers. So let me now pull up the second. And the question is whether you sell organic produce retail or wholesale, both or neither, if you happen to be a organic or other grower. So please check one of those boxes, and just another couple seconds and we'll pull up the third one, and this is for everyone in the audience of whether you buy organic fresh produce. So you can click yes, always or sometimes, or no, and we'll just have that up for another couple seconds, and we will close that and then share the results here. So yes, there are many consumers of organic produce here in the audience. So with that, let's move on to Sadhana Ravishankar. Sadhana, are you there? We don't quite hear you.
I'm here, but I'm not able to move to the slides. I mean.
Okay.
Hold on.
Okay. Sometimes there's a delay for a second or two.
Yeah, I'm not able to move the.
No? Okay. Just try one more time, maybe using the arrow key again.
Yeah, I use the arrow key and it doesn't
and it won't work. Okay.
Seem to move to the presentation yet.
For you. John, can you
click the
advance?
screen one time?
Okay. Just try clicking the screen one time again.
Yes,
I got it.
Excellent.
Yeah. Okay, thanks.
Yeah. Thank you. Thank you, Alice, for the nice introduction. And welcome you all to this webinar today. I would like to thank you all for taking the time to be here today to listen to my presentation. And in today's webinar, I'm going to be talking to you about a bunch of the work that we have done as a part of a big grant funded by the USDA NIFA Organic Research and Extension Initiative. And I would like to thank the funding agency for funding this project. So most of the work I'm going to be discussing with you today would be a part of a large grant. And in this particular grant, we have looked at all the way from attachment and internalization of foodborne pathogenic bacteria, irrigation water, to the post-harvest sanitization practices for leafy greens. And the collaborators in this project include the list given in here. Apart from scientists at the University of Arizona, we also have scientists from the USDA ARS Beltsville, Maryland facility, as well as Albany, California. And we also have scientists from the Oklahoma State University as well. Some of them are pictured in here when we took a tour of the Yuma growing region. There is a picture of some of my collaborators. And before we begin, I do have a question for the audience. If you could please quickly give some answers, then Alice will be able to read some of the answers. So the question here is, what are some of the significant fresh produce safety issues that you are facing currently? What are some of the significant fresh produce safety issues that you are facing currently?
Okay, yeah, so if you could just, if there are members of the audience that could type in some of the significant fresh produce safety issues that you face and some of the challenges that you face. I'll read a couple of them out loud. We have one answer so far, which is FSMA regulation compliance, and food safety for school garden to cafeteria, well water for washing. I'm a consumer, not a producer, but I don't buy organic leafy greens due to E. coli concerns, somebody says. Pests, selling to restaurants, and don't know how to adequately clean fragile produce. So those are some of the answers that we've gotten. Oh, one more.
All right, Alice. So do you think I can go ahead and proceed?
Yeah, I do. We just got one more, which was that it's expensive for small growers to comply. So yeah, you can go ahead now.
Okay. Thank you, Alice. So now coming to an overview of my presentation, I think I will be addressing some of the some of the issues that were mentioned by some of the growers during this poll. So coming to an overview of what I will be covering today. So this webinar is going to be in two parts. One is pre-harvest fresh leafy green safety issues, and the other one would be safety issues concerning post-harvest with regard to leafy greens. And what I have done is, we have from this grant, I have tried to address some of those issues which I think would be of most concern to you and would be most useful to you, and hopefully you will be able to go home with some messages that you may be able to apply in the real world scenario. So coming to pre-harvest leafy green safety, I will be addressing 4 different topics today. One is compost as well as compost amended soils, and we will be looking at what type of composts and compost amended soils may be better to use when compared to some of the others, and which are the ones that do not promote the survival of foodborne pathogenic bacteria. And this is a very important topic because compost is very commonly used by many of the organic growers. The next one is compost teas, and compost teas is becoming popular among many of the organic growers. And the belief here is that because all the nutrients and organic matter and the microbial burden is released into the water, these nutrients are going to be readily and rapidly available to the growing crops and will benefit the yield and quality parameters. So many of the organic growers are tending to use compost teas. So we will look at the safety of these compost teas. Then we will move on to animal feces. Animal intrusion is one of the concerns being mentioned to me by many of the growers in Yuma. So today we are going to see the survival of foodborne pathogenic bacteria in different types of animal feces. How long do they survive, and what will be of most concern to you, and so on. I will cover on that. And then finally, I will talk about coring tools. Coring tools are type of harvesting equipment that are used to core the lettuce that is used for ready-to-eat fresh cut produce industry. And coring tools, if they happen to be contaminated, they can transfer the contamination to the lettuce that are cored consecutively. So what we have done is we have looked at the potential of cross-contamination of different types of coring tools. We have come up with some modified designs as part of this project, and we are going to be looking at what is the design that would reduce cross-contamination from one lettuce to the other. So I will be describing to you about that as well. So that is on pre-harvest leafy green safety. Now coming to post-harvest leafy green safety, my lab has been working on a number of natural type of antimicrobials for the last several years, and we have tested a number of these antimicrobials in the wash water for washing leafy greens, and so we will be going over some of those. And we have also tested the efficacy of these antimicrobials during recycling, when you recycle the wash water, and we have some very good candidates in that, so we'll be discussing that. And then I've been working with a number of organic sanitizer manufacturers. They've been kind enough to supply me with their products, and we have done some work with that. So I will be discussing some of the organic sanitizers that have been as effective as the currently used chemical types of sanitizers and so on. And then finally, I'll talk to you about edible films. And edible films is a novel way of applying these antimicrobials in bagged salads. For example, some of these essential oils or plant extracts can be incorporated into any kind of a plant pulp. For example, you can make a pulp from a flower or a fruit or a vegetable and so on. You can include the essential oil in there. You can cast a thin film, and this film can be added as an ingredient into your salad bags. So we will talk about the efficacy of these edible films as well. And then finally, I'll talk to you briefly about the outreach activities that we have been doing at the University of Arizona. And Dr. Kurt Nolte, who is the extension lead in this project, is here today, and he may be able to answer some of the questions with regard to outreach activities. So let's go ahead with the presentation. Before we begin, I wanted to talk to you about what are some of the sources of contamination that can contaminate your leafy green in the field, especially if human enteric pathogens are going to be present. So one of them here is untreated manure. And in many farms, they have their composting facilities side by side with the field. That's not going to be in a huge difference or huge buffer zone in between the composting area as well as the field crops. So if these are not composted properly, what can happen is if human enteric pathogens are present, they can be dispersed by air or wind, and they can contaminate your fresh produce. And there has been some research done in my lab, and we have shown that dust can be an agent of transferring enteric pathogenic bacteria to produce. And then insect vectors. If you have insects, especially if you have a manure pile, can you keep away the insects? No, it's not possible. So these insects can carry foodborne pathogenic bacteria, and they can contaminate your fresh produce as well. And then you have other types of microorganisms present in the soil, such as fungi, protozoans, as well as nematodes that can transfer the contamination as well. And to begin with, if your seed is contaminated, it's going to be hard to avoid contamination. And then we have issues such as contaminated irrigation water. If the irrigation water is contaminated with the enteric pathogens, then you can have a large crop that can be easily contaminated. So the reason I'm showing this slide is we have tried to address some of these pre-harvest food safety issues, such as compost, compost teas, animal fecal material, as you can see in this, if you have animal intrusion, and let's say the fecal material has foodborne pathogenic bacteria, they can be easily transferred to your crop as well. So some of these we have addressed. We have done some work with contaminated irrigation water as well, or survival of pathogens in irrigation water, but due to lack of time, I will not be able to go over those results with you today. So let's look at some of the pre-harvest food safety issues initially. So the first one here is compost as well as compost amended soil. So in this part, we are going to see what type of compost may be better to use in the field compared to the others. And this work was done by my graduate student, Jennifer Todd. And she used 13 different types of compost, as you can see in the slide. And the main ingredient differs in these composts. For example, we have dairy manure-based compost, we have forest products, we have steer manure-based composts, and we have horse manure-based, poultry manure-based composts, and so on in here. And these are, I think, product 1 through 8 are commercially available composts, and the rest are proprietary formulations. So I'll be discussing the results mainly from the commercially available type of compost in here. So what we did is we directly inoculated these different types of composts with foodborne pathogens of concern to the leafy greens, and those are mainly Salmonella as well as E. coli over 5787, the two pathogens of most concern with regard to leafy green safety issues. And we incubated these at room temperature, and we looked at the survival of pathogenic bacteria in these cases. I'm not able to move, the slide is not moving forward. John? I'm not able to move it.
Okay, the slide has moved to the next one. So I don't know if you see it. It says survival of Salmonella Newport.
Yeah, she moved it. I don't know what was, maybe this was stuck. Yeah. So what you're seeing here is the result of survival of E. coli 015787 in some samples of compost as well as compost amended soils, and I'm showing you the commercially available composts. And as you can see in here, oh, there's supposed to be one more slide before this. I'm sorry. Oh, where is the slide before this? Yes, we got there. So this slide shows you the results of Salmonella in the various different types of composts, and I've only selected those samples that did not promote the survival of Salmonella so much. And as you can see in here, compost 4, 5, as well as 13, you see the survival of these bacteria only up to day 7 in case of compost number 4, and up to day 3 in case of compost number 5, and day 10 for compost 13, which is one of the proprietary ones. And these number 4 as well as number 5 are basically steer manure based type of compost, and these are the ones that we saw that did not promote the survival of Salmonella so much. And the next slide, please. This one shows the survival of E. coli 015787, and as you can see in here, E. coli did not survive as well as Salmonella did. And so here the sampling is basically hourly when compared to the number of days. And again, compost 1 through 5 are the ones that did not promote the survival and are shown in here. For example, if you take compost 1, 2, 3, 4, and 5, almost all of them, within 72 hours, the organisms were below detection, and especially compost number 3, the organisms were below detection even within 8 hours. And again, if you see compost number 4 and compost number 5, which are steer manure-based composts, they did really well as well. Number 3 is a combination of poultry manure and some other types of products, and that type of compost did really well. So looking at both these microorganisms, next slide, please. What did we learn from this part of the study? If we look at both the types of microorganisms, we clearly saw that the steer manure-based compost did not promote the survival of both the pathogens very well, and this may be a better option to use in the field when compared to the other types of compost. And there are two other things with regard to these composts. One is both these composts were much drier when compared to the other types of compost, and also with regard to the pH, they had a much higher pH when compared to the other types of compost, pH 9 or above, and both these conditions might have contributed to not being so much favorable for the survival of pathogenic bacteria. And in general, what we saw was Salmonella did survive in these composts for much longer when compared to E. coli 015787. We saw that there was better survival in that case. So samples 4 and 5 again may be better to use in the field. And another result from this study is, one might think that if the presence of background microflora is high, then compost may not promote the survival of pathogens, but we did not see such a correlation in our study. For example, sample 6 had the highest amount of background microflora. We saw about 9 logs or more background in that particular type of compost. However, it did promote the survival of pathogens. So there was no such correlation seen in this particular study. So the take-home message here is the steer manure-based compost, according to this study, may be better to use in the fields. Next slide, please. So now we move on to the compost teas. And so again, same type of question, what type of compost teas may be safe to use in the field conditions? And again, compost teas are basically made by steeping the composted manure from different sources. It goes into the manufacturer's instructions give you the information that you basically steep the tea bag in water, you leave it for like 24 to 48 hours. All the nutrients, the organic matter, as well as the microorganisms are going to be leaching into the water, and then this is applied to the crops. And many farmers like this because it may be an easy release of nutrients for the plants, and the plants will be able to easily and rapidly absorb the nutrients, and it will beneficially affect the plant yield and quality and so on. So in this particular part of the study, we looked at what type of compost teas are going to be better to use in the field conditions. And my graduate student Jennifer Todd did this work. We used 6 different types of compost teas, and the main ingredient here is either alpaca manure based, or cow or sheep or horse or poultry manure based types of compost teas. And this picture shows the ways these were prepared in the lab. And if you are wondering what an alpaca looks like, this is the animal given in this particular picture. Next slide, please. So what we did was we inoculated these compost teas prepared in the lab with the pathogens of concern, including E. coli 015787, Salmonella, as well as Listeria. And we studied the survival over a period of time. And what you are seeing here is how long does Salmonella survive in the various compost teas that we studied. So as you can see in here, the survival is up to 98 days for these compost teas. And the one that did not promote survival the best would be alpaca type of compost tea. Within 84 days, no survivors were detected. However, in the case of other types of compost teas, such as the sheep manure or horse manure, cow manure, as well as poultry manure based types of compost teas, the survival was still there after 98 days as well. So looking at this particular result, one might think that the 90 to 120 day regulation of application of these composts and compost teas may be better to follow because these organisms may be able to survive. Next slide, please. So next we will look at the results of E. coli 015787. And E. coli did not do as well as we saw as in the case of Salmonella. In this case, as opposed to Salmonella, in case of Salmonella, alpaca was the best one. It did not promote survival. However, in case of E. coli, alpaca was the only one that survived for more than 70 days. All the others, such as the cow manure based or the sheep or the horse manure based, no organisms were detected at 21 or 28, or even with the poultry manure based and another cow manure based compost teas, by 42 days no survivors were detected. So in this case the results were slightly different. However, E. coli did not survive as well as Salmonella did. So if you take out of the 90 to 120 day deadline of applying these before harvest, I think these should work for E. coli according to this the result you're seeing here. Next slide, please. So next we are going to look at the survival of Listeria monocytogenes, which is another foodborne pathogen of concern in many different types of foods, including ready-to-eat products. And the survival of Listeria was even worse when compared to E. coli. It did not survive as long as E. coli did. With the alpaca-based manure, the survival was only up to 35 days, and all the rest of them, the sheep, the horse, the cow manure-based compost teas, within 21 days, and the poultry manure-based, I think, was up to 28 days, and so on and so forth. So again, this organism also did not survive very well in compost teas beyond a month or so. So next slide, please. So now that we have seen that these organisms were surviving for a particular amount of time, we wanted to apply some of the compost teas in the field conditions and see what was going on, what the survival was. Was it similar or was it different? So we did this particular experiment at the organic plots at the Yuma Ag Center. And we used non-pathogenic E. coli in this condition because of course, you cannot use pathogens under field conditions. So we used a non-pathogenic E. coli. We prepared the compost teas, which were inoculated with the non-pathogenic E. coli, and then they were applied in the organic plots where we were growing leafy greens. And we used two different types of applications. One was a foliar type of an application. As you can see here, our team member from Yuma, Ramiro, is applying this using a backpack sprayer. And the other type of application is a side dress type of an application, which I will show to you in the next slide. In the side dress application, you have an equipment, as you can see in here, and there are blades that would keep injecting the compost tea containing water as you go through the field, and you will avoid adding anything foliarly. So previous slide, please. So we did apply using these two different modes of application, and we also did with and without fertilizers. In one batch, we applied the fertilizers, and in another batch, we did the experiment without the fertilizers. And then after application, we harvested the leaves at various time intervals, and these went through an enrichment process and plating on Petri films for E. coli. And the time intervals we used was 1.5 hours after application, that is the 0-day sampling, and then 3 days, 7, 13, 14, and 20 days, and so on. Next slide, please. So we did collect a lot of data, and what I'm going to show you today is just a few sample and then maybe some take-home messages, because it's a lot of data to discuss. So what you're seeing here is a side dress application of the compost tea on the iceberg lettuce. And here you see with the fertilizer what was the survival, and without the fertilizers what was the survival. And here you see Natalie as well as Janine, who are our Yuma team members who contributed to this part of the work. So looking at, and we did this samples up to day 7, and we included deionized water uninoculated as our control, and then we applied the compost teas by themselves, and along with E. coli, and then E. coli by itself as well, with and without the fertilizers. And with regard to field conditions, it's really hard to control what is going on in the environment. So the results were kind of variable, and as you can see in here, between day 0 and day 3, in some cases there was reduction, and in some cases we saw some survival. However, by day 7, we were not able to see organisms surviving in most cases, except just a couple of these compost teas. Now coming to the results without the fertilizers, again a similar trend was seen between day 0 and day 3, but with day 7, we were not able to see any of the survivors detected in this case. So in general, between the two types of applications, the side dress as well as the foliar type of an application, side dress was the mode of application which did not promote the survival of E. coli when compared to foliar type of application, and that might be a better way to apply these compost teas. And another finding would be that in general, when we applied these compost teas along with the fertilizers, there was better survival of bacteria compared to when we applied these compost teas without the fertilizers. So it might be better to go for applying these without the fertilizers, and a side dress application might be the way to go. Next slide, please. So the take-home message here is side dress application may be a better one than foliar application. So now we move on to the next one, which is animal intrusion. And to address this issue, we obtained different types of animal feces from Yuma, and we artificially inoculated those with the foodborne pathogen of concern, Salmonella as well as E. coli 15787. And we incubated these samples under different temperature as well as relative humidity conditions. For example, we used a low temperature, high relative humidity, and slightly higher than room temperature, low relative humidity type of conditions in this case. And we sampled cow, dog, as well as bird type of feces, and all these were obtained from Yuma. And among the 3 types of animal fecal material, dog feces was the one that did not support the survival beyond 7 days. And what we saw in here is low temperature, high relative humidity conditions did promote the survival better when compared to 26 degrees C, 40% relative humidity. So the survival was only up to 7 days. So it is possible that if you see a dog walking around your field, you may not need to worry too much about it. The next is bird fecal material, and looking at the results for the survival in the bird fecal material, Salmonella did a lot better than E. coli O15787, and low temperature, high relative humidity condition did promote the survival of Salmonella. Maybe it did survive more than 80 days. We did not do any sampling beyond 80 days. And now looking at the cow fecal material, the survival was the highest. For example, we saw survival for more than 100 days. And in this case, what we saw is the 26 degrees C, 40% relative humidity gave survival for even more than 200 days. And in case of both E. coli as well as Salmonella, compared to 15 degrees C, 80% relative humidity. So looking at the survival in animal fecal material, cow feces is something you definitely need to worry about. So if you see cow type of fecal material in your field, we probably need to worry about contamination occurring in the adjacent rows of crops and so on, especially if you have a lot of wind in the field. So next slide, please. So the take-home message from here is that pathogens do survive in differently in different types of animal fecal material, and it will be interesting to sample some of the other type of wild animal feces such as coyotes and javelinas and jackrabbits and so on. So now we move on to the next one, which is coring tool. And one part of the project, we looked at some of the modified designs. And in the proposal, we have mentioned that we will come up with our own coring tool designs, and we will compare that to the original coring tool design. So Dr. Kurt Nolte and Dr. Mark Siemens at the Yuma Ad Center, they designed 3 different types of modified coring tool designs. And we looked at these modified designs in comparison to the original tool design to see what is the cross-contamination potential when we use the contaminated coring tool to core like hundreds of lettuce consecutively one after another. So in this particular experiment, we did inoculate the coring tool using a non-pathogenic E. coli. And after drying this coring tool, we cored about 100 heads of iceberg lettuces. And after coring, we used this cored region, this area, which is more prone to contamination. We took like 10 gram samples from these, and we did dilutions as well as plating on Petri films to see which ones were positive or how many lettuce heads were positive for E. coli. And here you see our Yuma team, Diana as well as Emily, and also Ramiro, who helped us with coring the iceberg lettuces. So next slide, please. Let's look at the various designs as well as the results we saw here. So the modified design consists of 3 different types. And what you are seeing here is the original coring tool design. It does have a welding on the back, and it has a straight edge with the coring knife. And this shows you the design one. And the design one modification here is the front is slightly short, and the coring edge is going to be straight at the bottom. And what you're seeing here is design 1 length 2, which was the best design. And then this one shows you the design 2. And here you have, just like the original coring tool, the front is similar to the original coring tool, and then the edge is going to be a straight edge at the bottom. And what you're seeing here is design 2, length 2, which is 10.5 centimeter. And this one shows you the design 3. And in case of the design 3, you have not a very short front, but a regular front. However, the bottom edge is going to be at an angle in the front, as you can see here, slight angle in this case. And this one is showing you the design 3, length 3, which is like 13.5 centimeters. So we tested all these 3 combinations of designs, and this particular graph shows you what were the % of positive lettuce heads out of the 100 lettuce heads that were cored with the various designs. And looking at this, the best design that reduced the cross-contamination or had the least number of positive heads is design 1, length 2, where you, as you can see, it's only 30% positives in this case. And the next best design is design 2, length 2, followed by design 3, length 1. However, the original design had a very high number of positives, and it had, like, possibly close to or even more than 90% positive heads. So this graph clearly shows us that the modified designs are going to be better to use in the field in order to avoid cross-contamination in case, if one occurs in the field conditions. And we've also seen that with the original tool when we have done some studies with the bacteria that this welding region is the one that is more prone to attachment by bacteria, while this welding region is absent in many of these coring tool designs. So the take-home message here is that some of the modified coring tool designs are going to be better to use in the field in case if you want to avoid cross-contamination occurring in the field. Next slide, please. So this concludes the pre-harvest leafy green safety issues. Now we are going to move on to the post-harvest safety of leafy greens. And in my lab, we have been testing natural plant type of antimicrobials for inactivating foodborne pathogenic bacteria in different types of foods. And in case of leafy greens, what we did was we applied these plant type of antimicrobials in the washed water. And I'm going to be discussing with you some of the advantages of these plant antimicrobials compared to some of the existing chemical types of washing methods. One is that these antimicrobials are all generally recognized as safe. They are all of plant origin, and they not only kill the pathogenic bacteria, but they can also kill many of the spoilage type of microorganisms, which mean you will have an improvement in the shelf life as well as acceptability. And all these are being natural. They are clean label type of technologies. And we have done some studies to show that the efficacy of these natural plant type of antimicrobial washes is not affected by organic matter, which means there is a possibility of recycling the wash water. And as you will see in the results later, we have done recycling 5 times. We used the wash water 5 times, and the efficacy of some of these plant extracts remained intact. So they are pretty good for recycling. And then because these are biodegradable, they are going to be very environmentally friendly. And as opposed to chlorine, where you may need hot water, these are going to be more energy efficient because you can do the treatment under cold temperatures. You do not need any hot water for this. And another very important point to note about these types of antimicrobials is that they do have a residual type of an activity, which means they will continue to act over time. So let's say you have a cross-contamination event occurring post-processing, during packaging or after packaging, and if you have these antimicrobials, they will continue to act up to 3 days, 4 days, and so on and so forth, and they will be able to kill the bacteria present on your leafy greens. Then another advantage is that they can help in enhancing flavor. For example, oregano oil. Oregano is a herb, and the essential oil obtained from the herb oregano is already used in salad dressing. So there is a possibility that some of these compounds may help in enhancing the flavor. And being plant type of compounds, they also have other types of health benefits. For example, some of them have antioxidative, which means they may be anti-cancer. Some of them can be anti-diuretic. They can help in reducing cholesterol. Some of them may be anti-diabetic, and so on and so forth. So these are some of the advantages with regard to the plant type of antimicrobials, comparing some of the chemical types of washes that are currently used by the food industry. Next slide, please. So what we have done is we have used essential oils, plant extracts, as well as organic sanitizers, and looked at the efficacy of these in killing pathogenic as well as spoilage types of microorganisms. And with regard to plant extracts, we have used different types of extracts. For example, we have olive extract. Olive extract is made from freeze-dried olive juice. Apple extract can be made from the skin of apples. And then we also have hibiscus concentrate, which is made from hibiscus calyces. And we have looked at these plant extracts in comparison to hydrogen peroxide, which is approved for organic use. And what we have seen is the activity of these plant extracts can be even better than hydrogen peroxide. It's equal to or better than hydrogen peroxide. We got up to 3 logs reduction, which means you're able to kill more than 1,000 microorganisms per gram of your leafy green product. And we also saw that the activity with regard to hydrogen peroxide is one time. You use the wash water, and let's say there's like a half a log reduction over the storage, you're not going to see any more reduction. But with our plant antimicrobials, you will continue to see an increase in the killing effect of these pathogenic microorganisms. And with regard to background microflora, we were able to see up to 2 logs reduction just by washing. However, when we use these plant type of extracts, we were able to see up to 3 logs reduction with these plant extracts. And later you will also see that we have applied some of these plant extracts in recycling the wash water, and we have seen that the efficacy was remaining intact. They did not lose their efficacy with up to 5 washes of organic leafy greens in the wash water. Next slide, please. So what you're going to be seeing in this particular slide is the result from an essential oil, which is oregano oil. And this part of the work was done by my grad student, Catherine Moore. And what you are seeing here is we did a 1 minute as well as a 2 minute treatment. And the storage was at 4 degrees C as well as 8 degrees C. And we used 3 different concentrations, 0.1, 0.3, as well as 0.5% of oregano oil. And with 0.3 and 0.5% oregano oil, you can see that is complete killing. We did not see any survivors with any of these storage or exposure time conditions. And with 0.5%, even immediately upon exposure, we were able to kill the bacteria completely. So you can see how effective some of these essential oils can be. Next slide, please. So the next one, what you're seeing here is a biophotonic imaging of oregano oil as well as cinnamon oil treated romaine iceberg lettuce leaf. This work was done by my postdoc, Dr. Dev Kumar. And what you're seeing here is the leaf of romaine. And as you go up in the color intensity, the population of microorganisms is going to be more. Starting from blue all the way to red, you have higher population of microorganisms. So before the treatment, you're able to see some of the yellow regions and maybe red spots here and there. So there is high population of microorganisms. I think he did inoculate up to 10 to the 6 or 7. And after treatment, you do not see any glowing at all. And we used a bioluminescent salmonella for this, which contained the Lux gene. And using the Lux gene, it is able to glow. And as you can see in this after treatment, the leaf is still there, but you don't see any glowing, which means all the microorganisms have been killed during the treatment. So this wasn't the case of oregano oil. Now coming to cinnamon oil, again, you can see some hot spots in here where you have some yellow regions, some green regions, and some red regions as well. And this is before treatment, and after treatment, you can see that some of the glowing is already gone, and that shows that there was a killing of the bacterial population in this case also. Next slide, please. And this one shows a confocal scanning laser microscopy image of a fluorescent Salmonella Newport on baby spinach leaves that was treated with oregano oil. And this is the image before treatment, and this is after treatment. So before treatment, you can see all these fluorescent salmonella, and you can also see this stomatal opening, and many of the organisms are kind of concentrated towards the stomatal opening. And after the treatment, you can hardly see many of the fluorescent salmonella. The population has reduced considerably, as you can see in here, which shows that oregano oil was very effective in killing the salmonella population on the surface of baby spinach in this case. Next slide, please. So the next, we are going to be looking at a spoilage type of microorganism, and Pseudomonas is one of the predominant spoilage organism that can be found on leafy greens. And I'm just showing you a sample table here wherein we used essential oils on the surface of baby spinach, and we did hydrogen peroxide as well as phosphate buffered saline as controls as well. And to begin with phosphate buffered saline, we had about 4 logs in this case. And if you look at the efficacy varies between the essential oils, and some of them are even better or equally comparable to hydrogen peroxide. And one of them, the candidate essential oil A, with 0.3 as well as 0.5% concentration, we were able to see by day 1 there were no survivors detected. It should be less than 1 in this case. So it was very effective on inactivating Pseudomonas. And in other cases, we saw the efficacy was pretty similar, or in some cases maybe even better than hydrogen peroxide. And again, spoilage organisms are, especially Pseudomonas, is very difficult to inactivate with any kind of a treatment. So some of the essential oils are being pretty effective in this case. Next slide, please. So next, what we did is we wanted to do some combination treatments. We wanted to go one step further because one of the concerns with regard to some of these essential oils is the pungency. So if it is going to be really pungent, do you think consumers are going to like it? So that is one concern that we have to address. And so we wanted to see if we combine essential oil at a very low concentration with a plant extract which is not so pungent, and we try to see if we can avoid some of the questions with regard to the organoleptic attributes or the sensory attributes of the leafy greens, because our consumers going to like it. So we did some combination treatments, and this work was done by my grad student Zeroy Rada. And what you are seeing here is the results for the 4 leafy greens, and we did a combination of 0.1% essential oil and a 3% plant extract. And 0.1% essential oil is a very low concentration, and to begin with 0.1% concentration of essential oil, we were not able to kill Salmonella. However, when we combined this with the 3% olive extract, we did some better reduction. So combination treatments proved to be much better than individual treatments. For example, we did see some synergistic effect in case of romaine lettuce. We had like 3.5 logs reduction, which is much better than many of the currently used sanitizers. And some additive effect was seen in case of baby spinach, as you can see in here, 3 logs reduction and so on. With the others also, we did see, compared to the individual treatments, combination treatments definitely gave us better reductions. Next slide, please. So next, we wanted to go one step further. We wanted to see what is the efficacy of these antimicrobial washes when we recycle the wash water. And this research is very important for the fresh produce industry because recycling is a practice that is very commonly used by the produce industry, and it is a good economical saving for them if they will be able to recycle the wash water. So what we are seeing here is results from one of the essential oils, one of the plant extracts, and also the phosphate buffered saline control that we used in this case. So we began with 5 logs population, and with the PBS control, you can see anywhere from 4.1 to 4.4 logs. So there was less than a log reduction just with PBS, maybe like a half a log or so on. And coming to the essential oil, with the first wash, we were able to see no organisms detected. It was very effective, and the effectiveness continued throughout the three days of storage. And as we did the subsequent washes, there was slight decrease in the efficacy. However, the counts were still lower than the control that we used. Now coming to the 3% plant extract, what we saw here is about 3 log survivors during day 1 with the first wash, and we saw more reduction as we went by day 1 as well as day 3. And if you look at the 5 washes, between the first wash and the 5th wash, not a big difference. That shows that these plant extracts were able to maintain their efficacy with 5 washes, not only day 0, but also by day 1, not a big difference in the counts. And again at day 3, not a big difference there. We began at 2.4 and we are ending at 2.4. So this shows that the efficacy of some of these plant compounds can be maintained by 5 washes. And this is the result for organic baby spinach, and we had inoculated Salmonella on the organic baby spinach. So this shows that these can be pretty effective with recycling as well. Next slide, please. So next, you will see what happens to the wash water. So what we thought was, okay, we do see organisms on the leaves. So is the wash water contaminated? So what we did was we sampled the wash water after each wash. And what you're seeing here is the PBS control population in the PBS control wash water. And then the wash water containing 0.3% essential oil and the wash water containing 3% plant extract. And as you can see with the PBS control, the population increase began at 3.6 and it kept increasing with the 5th wash, showing 4.3 logs. However, in the case of essential oil as well as the plant extract, boom, no organisms detected. As you can see here, the water was pretty clean. So this shows that both the essential oil as well as the plant extract can be highly effective in killing the microorganisms. So this shows that these are definitely potential candidates for using in wash water and recycling that same wash water. So this photograph shows my lab manager, Libinzu, and grad student Arlette Schneider. And these are some of the huge tanks that we have been using in our recycling experiments. And you see Iceberg lettuce. And here is my postdoc, Dr. Dev Kumar, and my grad student Zeroy doing some treatments in the tank as well. And this shows you the wash water after washing 5 times of the Iceberg lettuce. And you can see the water sounds not so turbid as you would expect it to be. And this part of the work was done by my lab manager, Libinzu. So the take-home message here is these antimicrobials can be definitely good candidates for using as well as recycling the wash water. Next slide, please. So now we move on to the organic sanitizers. And we have tested about 10 different organic sanitizers. And I'm showing you the topmost candidate, which is Chico Wash. And Chico Wash is manufactured by E3 Organics based off of California. And we did treatments of 1 minute as well as 2 minutes. And we used Salmonella on the leaves. We also compared our results with the hydrogen peroxide treatment, 3% hydrogen peroxide treatment. And as you can see in here, even these organic sanitizers did have activity increasing during storage. With hydrogen peroxide, you see anywhere between like a half a log to up to 1.5 logs reduction. And in most cases, you do not see an increase in activity during the 3 days of storage. However, with the Chico Wash, and Chico Wash is basically a citric acid based type of a sanitizer, and you can see that with both 1 minute as well as the 2 minute treatment, we were able to see an increase in residual activity during the storage, and we obtained up to 3 logs reduction in some cases, both for Iceberg as well as baby spinach. And as you can see in this table, Chico Wash was much better than just hydrogen peroxide in inactivating Salmonella. So pretty good reductions in this case. And we've also done some recycling experiments with the Chico Wash, and we have been able to show some pretty good reduction. And the good thing about this sanitizer is that the pH is very well maintained. It has a very low pH, and the pH is maintained. Even if you do like 50 washes, the pH doesn't go down. It stays from, it may change from like pH 2 to up to 2.2 or something or so on. And due to lack of time, I'm not able to discuss all the results. However, this organic sanitizer was top in the list of the sanitizers we have tested so far. Next slide, please. So now we will talk about the edible films. So edible films, as I told you, is a unique way of incorporating these plant-based antimicrobials into films that are edible. And the reason these are called edible because these can be made from any plant part. As long as you have a pulp, for example, a fruit pulp or a vegetable pulp, or let's say you have leaves, you can make these from spinach, tomato, carrot, or any type of plant-based material, hibiscus, flowers, these edible films can be made from. So what you're seeing here is films made from apple pulp. So apple pulp has been, in this case, incorporated with carvacrol, and carvacrol is the active ingredient in oregano oil. And here you're seeing a cinnamaldehyde-based apple film, and cinnamaldehyde is the active component in cinnamon oil. So what they basically do is they have the pulp, they add the antimicrobials and a few other components, they cast it into a very thin film, and then you can cut this film into any shape or any size you want according to the food product. You can wrap the food product with this film, or in our case with the leafy greens, what we did was we added them as ingredients in the salad bags. Okay. And the edible films are made at the Albany lab. My collaborator, Dr. Friedman, he is the one who makes these edible films for us, and we test them in our lab. So what we did do is we cut these edible films into small pieces, and we disperse them evenly throughout the salad bag. And these salads contain the leafy greens that have been inoculated with the bacteria. And then we store these at 4 degrees C for up to 7 days. And what we have seen is complete reduction of microorganisms depending on the type of edible film and the type of antimicrobial used. And again, carvacrol-based films have shown wonders. They can inactivate high population of microorganisms. So we have seen pretty good reductions with both Salmonella and also some of the spoilage organisms. We have seen some reductions as well. And as you can see in here, these come in plastic material, they are stuck to it, and then we start using them. And so you can make these films from anywhere, and the take-home message from the edible films is that they definitely are efficacious in killing the bacteria in your salad bags, and they can be consumed because they are made of plant material. For example, let's say you make edible film from carrot. Carrot are consumed as salad vegetables. Tomatoes are consumed as salad vegetables, and so on and so forth. Excuse me. So these are consumed as salad vegetables, so they can be used as ingredients. And as long as we can maintain the concentration of the antimicrobial to a level it is not so pungent, we should be okay. And again, we need to do some sensory studies with the salads containing edible films in the salad bags, and hopefully we hope to reach one day where these films can be used by the industry. Next slide, please. So this concludes the research with regard to the post-harvest aspects of leafy green safety. Now I want to talk to you very briefly about the outreach activities that have been done as a part of this project. And here you see Dr. Kurt Nolte, who is the extension lead in this project, and he is the director of the Yuma County Cooperative Extension. And he has done a number of outreach activities, and one of them is he has conducted several organic food safety workshops in various regions of Arizona. He has conducted more than 10 workshops, and he has trained more than 250 participants so far. And he goes over many of the important aspects of leafy green safety issues as well in these workshops. And then he's also done some trainings, tours, as well as demonstrations, and he has covered more than 1,000 participants in these trainings and demonstrations and so on. And then he's also developed some extension videos. More than 10 videos have been developed, and some of these videos, they do cover some of the topics that we went over today. For example, edible films, composts, compost teas, irrigation water quality, and so on and so forth. And if you go to this particular web link, you should be able to access these extension videos. And then most importantly, he has come up with a 4-part video series, field worker training modules. And in these, he has covered some important topics such as field sanitation, hand washing, field etiquette, and bodily fluid discharge types of topics. And these have already been adopted for use by many of the Yuma fresh produce industry, and we have got some very positive feedback from the people who have been using these videos. And he has developed this both in English as well as Spanish, and once these were developed in Spanish, we saw that the participant number grew up very well. And then he has also been using social media networking such as Facebook, Twitter, as well as Pinterest for the outreach activities of this project. And then a website called the Fresh Produce Safety website has also been developed. The web link is given in right here, and you can check it out. And many of the leafy green safety issues are covered in this website as well. And if you want to know more about the outreach activities, Dr. Kurt Nolte is here today, and he will be able to help you out today. Next slide, please. So that's all I have, and so let's go over some of the take-home messages from this presentation. What were some of the salient findings from what we covered so far? I know I talked about several different projects, so I want to go over these one more time so that you have some take-home messages with you. Hopefully, you will be able to apply some of these in the real-world scenario. So from the work on compost, we saw that steer manure-based composts may be better to apply in the field because they did not promote the survival of foodborne pathogenic bacteria. And coming to the compost teas, from our experiments, we have clearly seen that side dress application of compost teas may be safer to use in the field than foliar application. And not only that, it may be better to apply these composts without the fertilizers rather than with fertilizers, because without the fertilizers, the survival of microorganism was not as much as we saw with the fertilizers. And then with regard to animal intrusion, depending on the type of fecal material you have in the field, pathogens may survive for a long period of time. For example, in cow feces, we saw the survival for more than 100 or even more than 200 days, depending on the temperature, relative humidity of the growing conditions possible. So animal feces do carry pathogens and they can be harmful to your crop. And then coring tools, a modified coring tool design was definitely much better in reducing cross-contamination when compared to the original coring tool design that had the welding. And so if you're interested in using this modified coring tool design, you might want to contact Dr. Kurt Nolte, and he might be able to help you out in that. And then post-harvest sanitization practices, including plant antimicrobials as well as organic sanitizer washes, definitely kill the bacteria, and they have certain advantages compared to the existing practices in that they are environmentally friendly, they are biodegradable, they are energy efficient because you can apply them at cold temperatures, and most of all, they can be used in recycling because their efficacy is not affected in the presence of organic matter, as in the case of chlorine or some other types of chemical sanitizers. And then they do have residual activity in that they may help in preventing or reducing contamination that can occur after post-processing or post-packaging, because they maintain their activity during storage, and they can continue to kill the bacteria on your leafy greens. And similar is the case with organic sanitizers as well. They do maintain their activity. And antimicrobial edible films are definitely the candidates that can help in improving or enhancing the safety as well as post-harvest safety as well as quality of bagged organic leafy green salads during storage. So these are some of the take-home messages I can give you right away from the work we have done so far in this project. Next slide, please. So before I conclude, I would like to thank the efforts of my lab group. Without their efforts, all these results would not have been possible. I'd like to thank my lab manager, Libinzu, my postdoc, Dr. Dev Kumar, and all my graduate students, current as well as past graduate students, current as well as past undergraduate students for contributing to this work. I would also like to thank the UMA lab group for their help with the coring tool transfer studies as well as the field studies. Without them, we could not have gone this far. And I would like to thank all my collaborators from the academia as well as from the industry, such as E3 Organics, GTX Technologies, Cree Agri Incorporated, and also Biosafe Systems and all others who have contributed by sending their product for testing as part of this project. And most importantly, I'd also like to thank the USDA NIFA Organic Research and Extension Initiative, the Arizona Leafy Greens Marketing Agreement, Arizona Department of Agriculture, Arizona Iceberg Lettuce Research Council, and all the other industrial members that provided us with the funding to do our organic leafy greens research. I want to thank them all. And again, thank you, a big thanks to all the audience for taking the time to listen to this webinar, and I will be happy to take any questions now. Next slide, please. And here is my contact information in case you need to contact me. Thank you so much.
Wow, thank you very much, Sadhana. That was a really interesting presentation, and we definitely have a lot of questions coming in. I just wanted to mention that we did record this presentation, so if you'd like to view it again afterwards or share it with anyone else, we're going to be putting it on the eOrganic webinar archive and the eOrganic YouTube channel. And I also wanted to mention that we really value your feedback, so we'd very much appreciate it if you could fill up the follow-up survey, which you'll be receiving later today in an email. Also, you're welcome to use the eExtension Ask an Expert service if after the webinar you have additional questions about organic farming, and Sadhana very kindly provided us with contact information as well. So, moving on to the many questions that we're getting in. We had a few questions about the cost consideration. One person said that she's found that essential oils are very expensive. And so is it economic to use these oils, and how expensive would some of these treatments be?
Okay, coming to the essential oils, yes, they are expensive, but you will be using very little of it. And again, you will be able to recycle the wash water without a loss in the efficacy. So yes, they are expensive, but some of the costs can be offset by being able to continuously reuse it and by using very little of it. And some of the plant extracts, in comparison to essential oils, such as the apple extract, they are waste byproducts. Like apple skin is a waste byproduct of the apple industry, which let's say you are able to combine these essential oils and use it in combination with the plant extract, that might be a technology or that may be feasible and economical.
Okay. I guess there's someone who wanted to know whether carvacrol and cinnamaldehyde are approved for organic production right now, and whether those are substances that are on the national list.
Yeah, all these essential oils in the generally recognized as safe list of compounds, and so I believe these, because these are active components of these essential oils, they should be, because these are generally recognized as safe, they should be, I mean, there are, if you go to the organically approved list of components, I'm not sure if you will be able to find all of these, but because these are of plant origin and these are generally recognized as safe, they should be able to use it.
Okay. We had two questions from people wondering whether the compost teas that you used were aerated or aerobically brewed.
Okay, the compost teas that we used in the lab were not aerated. They were basically static. So we steep the tea bag in tap water, in deionized water for 24 hours. And during this process, we were not generally aerating anything. We were just using it as such. But aeration will, as I would know, would improve the growth of beneficial microorganisms. That's something that we have not tested so far here.
Okay. Let's see. This person has heard in a couple of separate presentations that leafy greens should not be washed in water more than 10 degrees colder than the greens to avoid pathogen internalization, but they can't find any science to back that up. Do you know whether this claim is accurate? And if so, do you have any, do you know of any research about that?
Okay, leafy greens, and so you mean lower than 10 degrees C. That has been, when it comes to internalization, it is a highly controversial topic, and previous research has shown that in some cases people have seen internalization, and people have not seen internalization under various scenarios. So it is very hard to come up with a conclusion that there is definitely going to be an internalization. But one thing that many papers have shown is that you need to have a certain population of microorganisms for them to be internalized. And they have seen that, especially for, I think, in case of fresh produce, you need to have, I think, at least 10 to the 5 or 5 logs of microorganisms in order for internalization to occur. And so I would think that, I mean, it's better to wash these leafy greens rather than not to wash them. And I think in the industry, the practice is to keep the temperature cold during washing. So, there is no conclusive evidence to show if there's definitely going to be internalization or not. And we are doing some research with regard to internalization in our grant, but we don't have any results yet on that. And that's something that we might want to research on, and I will definitely let you know what we find out, especially with water at cold temperatures less than 10 degrees C. Do you have a point? Yeah.
Okay.
Why don't
Thank you.
I'm giving the mic to my postdoc, and he has a point.
Hi, this is Dr. Ravishankar's postdoc. And for the question for water below 10 degrees, in tomatoes, they have shown internalization, or at least internalization of wash water inside the stem scar tissue when there's a temperature difference. And so usually for fruits like tomatoes or cantaloupes, that might be an issue. Thank you.
Thank you.
Yeah, thank you, Guru.
Okay. Let's see. Somebody wanted to know whether the compost teas and the manures were inoculated.
Yes, in our lab studies, we did inoculate the compost as well as compost teas with the pathogenic bacteria because we were studying the survival of these bacteria in the laboratory studies. And in the field studies, when we applied the compost teas to the field, we used both inoculated as well as non-inoculated compost teas. And obviously, we couldn't use pathogens under the field conditions, so we used non-pathogenic E. coli, which has similar survival characteristics to that of pathogenic E. coli 015787. So yes, in both cases, the compost teas were inoculated. And to begin with, what we saw is even in uninoculated compost teas, the E. coli was present to a certain extent. So that's what we had. Yeah.
Okay, this question I believe refers to the experiments that you did with washing salads and also the Chico wash. How do the counts of microbes that were in the results compare to, I guess, quote, safe thresholds? So in other words,
can you repeat that question again, Alice?
Yeah,
Sorry.
Somebody wanted to know how the counts of microbes, I mean, because not all of the washes completely eradicated the various microbes. So this questioner, I believe, wanted to know how the levels that remain compared to what is considered a safe threshold.
Okay, with our studies, what we had done is we had 5 logs of microorganisms to begin with on the leafy greens, and with the Chico wash treatment, we had close to a 3 logs reduction. So which means you're still going to have some organisms still surviving. And again, it all depends on who is consuming the leafy green. That it's going to be risky. Again, if it's an immunocompromised individuals, maybe it is risky because some of these organisms, even like 10 cells of E. coli or even 1 cell of E. coli can make you sick. So again, it depends on who is consuming it with regard to the risk. And the infectious dose for a normal healthy individual for some of these pathogens, for example, Salmonella, is going to be close to 10 to the 5. So what is the risk? Again, it depends on who is consuming it. But looking at the reduction, 3 logs reduction is definitely considered to be very good for fresh produce. Anything that gives more than a log reduction, because the currently used technologies give you 1 to 1.5 log, maybe at the most 2 logs reduction. So with regard to the reduction, this would be considered a pretty good reduction. I mean, obviously we want complete inactivation, but in some cases it may not be possible. And again, in the real world scenario, you may not be seeing 10 to the 5 or 6 microorganisms. To begin with, we may be seeing something like 100 to 1,000 organisms. And in that case, if we get a 3 log reduction, we possibly be maybe eradicating whatever is present on the surface of the leafy green. So we have to look at it in a comparative point of scenario. For the laboratory studies, we have to start with a certain level so that we want to see if maximum reduction can be achieved. So in order to get an enumerable level of population, we are forced to begin with a slightly higher level. But in the real world scenario, that may not be the case as such.
Okay, yeah, I guess we had a couple questions just about that terminology. Just if you could sort of say in lay terms what 5 logs of microbes means, because logs is not a word that a lot of people are familiar with.
Yeah, okay. 5 logs means 100,000 microorganisms. Okay, 4 is like 1,000 microorganisms. 3 logs is 3 logs is 1,000 microorganisms, and 4 logs would be 100,000 microorganisms.
Okay. We got another comment about the question that you asked earlier about some of the challenges and issues that people are facing with produce safety, and one of them was that food safety makes conservation practices difficult. And I know that's a concern for many organic growers. In kind of a related question, what are the risks from livestock urine? In other words, if I avoid visible manure, does that address the food safety risk?
Yes, visible manure in the field would definitely be considered a risk. And again, I'll let Kurt answer this question. I would think that visible manure in the field should definitely be considered a food safety risk, and they should try to avoid harvesting the adjacent rows of crops. Again, there is no guideline to say how far, how much you do not harvest, how many rows do you leave close to the animal fecal material being present in the field. Yes, visible fecal material will definitely be a risk. Kurt, do you want to answer?
Okay.
Can you repeat that question, Alice? I'm sorry.
Yeah. Okay. No, that was the question. The next question, I guess, back to the question of what a log is, like 100,000 microbes per what? Per a certain quantity of water?
Yeah, it is all our numbers are based on per gram or per mil of leafy greens.
Oh, okay.
So when we say 3 logs, it is going to be 1,000 organisms per gram of leafy green.
Okay, great. Thank you.
Yeah.
Okay. Here's a question about your coring tool experiment. What sanitation practices are currently in place in the lettuce processing industry to prevent cross-contamination by coring tools?
I think the recommendation is to use 200 parts per million of chlorine to sanitize these coring tools.
Okay.
Yeah, does that help answer the question?
I think
or
so.
do you want to repeat the question, Alice?
Okay, yeah, the question was what sanitization practices are currently in place in the lettuce processing industry to prevent cross-contamination by coring tools?
Coring tools, yeah, 200 ppm chlorine is what is used.
Mm-hmm. Okay, and then there's a question about whether there's a risk from cat feces as well. Or do you know of any
feces? Okay, that's an interesting question, and we have not tested any cat feces so far.
Okay.
Yeah, I think one parasite that people need to be concerned with regard to cat fecal material would be Toxoplasma. And Salmonella, of course, is present in all different types of animals, but not so much of other types of pathogens. So I think Toxoplasma as well as Salmonella is something that may be of concern in these fecal material.
Okay, we have time for one last question here, and that is someone commented that bacteria cells outnumber human cells 10 to 1 in our bodies, and it's clear that beneficial bacteria in the gut are essential to health. So are researchers measuring the extent to which these washes kill good bacteria on the greens?
Yeah, that's a very good question. And that's something, we definitely need to address in future research. We have not tested the effect of these sanitizers or organic antimicrobials against beneficial bacteria, but we do have plans of conducting this research in the future, specifically lactic acid bacteria. And speaking of that, more research needs to be done to see what happens in vivo when humans consume these. Let's say you have a contaminated piece of a leafy green, and you have treated them with these organic antimicrobials, and you consume them, or let's say you have consumed a contaminated piece of a leafy green, and you are consuming one of these antimicrobials as another food ingredient. What happens in vivo? What is going on with these antimicrobials? Can they kill just the pathogen selectively? We don't know. Maybe that is probably not possible, but that's definitely an area of research that we will be conducting in the future.
Yeah, thank you. That kind of answered the second part of the question too. Okay, well, we're running out of time, but I would really like to thank you, Sadhana, for this very interesting presentation, and I'd like to thank everyone online for your questions, and mention once again that you'll be able to find this and many of our other upcoming and archived webinars on organic farming and research topics at the link on your screen. And again, we'd also very much appreciate it if you could fill out the follow-up survey that you'll be receiving in an email later today. So thanks again, Sadhana and Kurt and everyone there in Arizona for chiming in. And thank you to everyone for joining us today.
Thank you, Alice.